Hey, good morning. Starting a new unit here today with today's notes. We are taking a look at Unit 4 today. So in this unit, we're going to look at proteins and carbohydrates. And we're going to start 4.1 with proteins. We've already seen several of them. They do a lot of jobs around the cell. We talked about a lot of proteins involved in DNA synthesis and translation and transcription. So we've seen a lot of proteins. Now let's take a closer look. So proteins So proteins are a macromolecule. We're kind of continuing our tour of macromolecules. Again, these are large molecules. I like to think of trains, big, long trains. And all of those macromolecules are made up of train cars that we call monomers. And in the case of proteins, the monomer is the amino acid. So I'm going to skip a couple lines here. Go down just a little bit to draw an amino acid. So all amino acids have this basic shape here. Where some of this hopefully looks familiar to you. This end here, do that again in blue, make it stand out. This is called a carboxyl group or carboxylic acid. We saw these guys when we did our lipids. So remember, each fatty acid had a carboxylic acid on top. So it's a carboxylic acid. Carboxylic acid is what this is. I've got the word acid underlined there. Let's take a look at the other side. Over here on the other side in red, this is what's called an amino group. I'm going to underline the word amino there. So there we are. That NH3 or NH2 sometimes, we'll see in just a second, is an amino group, a carboxylic acid on the other end, and that's why you can see it's called a carboxylic acid. So the name isn't just made up. Up on top, this R group, this R is like a variable, like X or N in math class. So it could be a lot of different things. So it's different for each amino acid. Sorry, I'm not very good at writing with colored pencils, but that R group is different for each amino acid. Again, like an X or an N in math, this is a variable. It can change. We have 20 different amino acids. The base part down here with the carboxylic acid and the amino group, they don't change. This is always the same. It's kind of like the sugar and the phosphate backbone of a nucleic acid. And then the only thing that changes is the nitrogenous base in the middle. Well, the only thing that changes in an amino acid is the R group right here. So that's different. That's why we have 20 different amino acids. All right, so we're going to combine this with another amino acid, create a chain. So just a little more room there. We're going to erase this O, and you guys will see why in just a second. And this N only has two H's on it. And the reason why, we're going to go ahead and connect right here, the carboxylic acid on one end, the nitrogen on the other. And so this right here, what also came out when we bonded these two together was, well, take a look at it. We had an OH up here on the carboxylic acid that's not there anymore. And we had an H on the nitrogen. So what came out was an H2O. So amino acids are linked through dehydration synthesis. Boy, we're seeing that again. We're seeing that again. Ah, man, I ran into the hole on the page. Terrible planning. So when that water comes out, that's dehydration synthesis. The water comes out, and that's what links right there. And this is a peptide bond. That link between amino acids is a peptide bond. We'd probably have another one over here. Color that one orange as well. Kind of have this trailing off in the distance, our longer chain, so we can kind of keep going. Not terribly concerned with it. There we go. This is our train. Again, this is just one car, one amino acid. There's another car, and they're linked together. And a big long linkage of them is our train or polypeptide. So these big long chains of amino acids are called polypeptides. And you can probably see why. Poly meaning many, and peptides are the bonds between them. So many bonds, many of these types of bonds gives us a long chain of amino acids. So there's actually a little bit left over from last chapter. These polypeptides get put together at a ribosome. So that's where they're linked. Again, that's what we looked at last chapter. Amino acids being linked together at a ribosome to make a polypeptide. And today we're going to go a little deeper, a little longer. So as they're assembled, proteins actually have a level, many levels, what we call folding. And ultimately, it's these folds that determine the shape of the protein. And this is super important. You're going to hear this a lot this chapter and beyond. In fact, you probably already heard a little bit. Shape equals function. Very, very important to have the correct shape so that the protein can do the job that we're wanting it to do. So after being assembled, the protein has several levels of what we call folding that ultimately lead to the correct shape of the protein, and that is super important. And that's actually what we're going to take a look at. There's a little AA for amino acids. There's a peptide bond between them. This is known as the primary level of fold. And this is the sequence of amino acids. So how they're assembled, how the genetic code says, hey, get this amino acid first, followed by this one, followed by that one, followed by that one. That's the primary level of folding. Now we'll take a quick look out just a little bit. Two of the same color together, because why not? All right, so got this polypeptide chain. So it's the same chain we were just looking at. We're just a little more zoomed out now. There's the polypeptides between it, but now we've also got connections between different amino acids. So they aren't sequential amino acids, and they're connecting. That's helping. You can see how that starts to fold back and forth, back and forth. So this is the secondary level of fold. And this is where we have non-consecutive, so not next to each other. Non-consecutive amino acids are going to link with different things, such as hydrogen bonds. We've already taken a look at some of those. We saw those in DNA. Also, it could be what we call a disulfide bond. Now, we're not going to spend a whole lot of time on that. It's very similar to a hydrogen bond, except with sulfur instead of hydrogen. So got these two different types of bonds. There are a few others. Some put etc. Here, there are a few other types of bonds, but not terribly important. The important part is that these amino acids are interacting with one another. They're not consecutive, but they're kind of joining up, and that causes these new folds that we see occurring on our protein chain, or polypeptide. That's going to bring us to our tertiary, and this is where folds kind of interact with folds. So, all right, there's our initial fold. Oops, camera here. So here's our initial fold, and we kind of came down and went out like this, back and down. So here's what we have. And I'm zoomed out way far. We can't even see individual amino acids anymore. And here's our hydrogen bonds or disulfide bonds, holding those two folds together. Well, oftentimes these folds kind of wrap around and fold in on each other again. And this is our tertiary level of folding. That's the tertiary level, and this is when folds interconnect with other folds. Finally, we do have one more level, although this doesn't happen for every amino acid. So here's the one we were just looking at. This was our tertiary folding, where folds kind of interlap with other folds. And now we're actually going to add another entire amino acid chain. So the green represents one chain of amino acid, one polypeptide, and the pink represents another polypeptide altogether. Now, this doesn't always happen. Sometimes proteins are done here. They only go to the tertiary level, but there are other proteins that go to the quaternary level. That's the fourth level. And that's when you have more than one amino acid. We got a pink amino acid chain, that's a polypeptide, and the green. And so multiple of them are going to interact to form the final protein. So again, it doesn't always happen that we get to the quaternary level, but a lot of proteins do. One of the more famous proteins, hemoglobin, I think has 4 different amino acid chains working in it. So quaternary levels, when you got multiple different polypeptide strains that all work together. All right, to finish these up, all right, proteins, kind of back out here. Proteins carry out a lot of different things in the cell. Wide range here. So proteins carry out a wide range of functions for the cell/organism. So not everything a protein does is just for the cell. Sometimes it's for the overall good of the organism. For instance, hair. The cells that are making my hair probably aren't getting a whole lot of benefit out of the hair, but they are, and that's proteins. So some of the things they do, they are in charge of recognizing pathogens. So those are basically any foreign body, whether it be bacteria or viruses. We're recognizing pathogens. They do a lot of cell-to-cell communication. They provide structure and support for the cell. We'll see in a few chapters that they are involved in transport and storage of goods. And probably the big one we'll focus on this chapter, they work on enzymes, work as enzymes, excuse me, to speed up the reaction. So that's what enzymes do. They speed up the reactions. I think you guys all have the homework. We can take a look at that in a few minutes here. One final note. And that's that 60% of a dry cell's mass is protein. So we all know that most of a cell is made up of water. The vast majority of a cell is water. Well, if we could dry that cell out, so no water, all we have left are basically the 4 macromolecules, probably 99% of the mass of a cell would be one of those 4 macromolecules, lipids, nucleic acids, carbohydrates, and proteins. Well, if you could dry out all the water, take out all the water, most of the rest of the cell, 60% of a cell's mass is protein. So proteins are incredibly important. They're bigger, they're more, I don't want to say more important. Obviously, everything's very important, but they're more abundant than all other macromolecules combined. 60% versus, and you guys can do the math. The other macromolecules are only 40%, all three of them combined. So proteins are quite important. All right, well, that finishes our 4.1 notes. So I'll see you guys here again next time. See you later.